Very good evening to everyone. I am Dr. Jayanti Saha from Siliguri, West Bengal. My college name is Acharya Prafulla Chandra Ray Government College. And today I have selected one topic from microbiology section, that is the life cycle of viruses. All of you know that viruses are intermediate between living and non-living, and they are obligate parasitic organisms self-replicating. And for their replication, the main material that is responsible is nucleic acid, either DNA or RNA. And I'm talking about the first virus that consists of the genetic material surrounded by the protein code. Okay, this is capsid. Okay, and this is the genetic material, basically DNA. As it is a first virus, then basically this is DNA. Okay, so this is the virus particle, and when this virus particle attacks a particular host species, suppose this is one bacterial cell. Okay, that is the host species. Okay, and after the first step, in the case of lactic cycle, the first step is attachment. Okay, and attachment, that means the penetration of these cell fibers onto the host cell. Okay, and basically some enzymes responsible for the formation of pore, and through this pore, viruses only inserts their genetic material into the host cell. Okay, so number one step is, we may say that this is actually attachment. Okay, attachment and penetration. This is the number one step. Okay, and number two, that means in the next step, what happens? The genetic material simply enters within the host cell. Only the genetic material enters. Okay, and upon the insertion of the viral DNA, the bacterial DNA actually is being degraded. Okay, this is the host cell. Suppose this is bacterial cell, and this is the bacterial DNA. Clear? On insertion of viral DNA within the host cell, the bacterial DNA is being degraded. Okay, and this is in the next step, upon insertion within the host cell, the viral DNA or viral genome suppresses the protein synthesis of bacteria, and rather it starts the synthesis of new protein particle of their own. So in the next step, after the degradation of bacterial DNA, the viral genome synthesizes the protein particle. Okay, that is the viral protein. Clear? Okay, and then simultaneously they also form the genetic material. Okay, genetic material particle of virus. Okay, and then in the next step, the mature first particles are being produced, consisting of the newly synthesized viral proteins and newly synthesized genetic material. Okay, and in the next step, the new first particle is being formed, comprising of the protein particle, that is the capsid. Okay, early protein, late protein, and the genetic material. Okay, within this. Okay, and after synthesizing the numerous first particle, they put a pressure onto the bacterial cell wall. Okay, and gradually they are liberated on the disruption of this cell wall. Okay, that means first of all they put a pressure on maturation, because the first particles then will be numerous. Okay, and they put pressure on the bacterial cell. Okay, and they liberate, they lyse the bacterial cell and deliver it and release. Okay, and this that is, I mean, in this way actually the first particle virus first particle is being liberated. Okay, and the whole process is actually the lactic cycle. Okay, and on the contrary, in case of lysogenic cycle, one extra important step is being carried out by the viral DNA. Okay, that is the prophase formation. Here, prophase formation. This is very crucial step, and that is not found in case of lactic cycle. This prophase formation is only found in case of lysogenic cycle. Okay, so what is basically the prophase? In case of lysogenic cycle, what happens once the viral DNA inserts within the bacterial or host cell genome, they integrate with the bacterial genome. Okay, so in case of lysogenic cycle, what happens? Prophase will be formed. Okay, and prophase, what is prophase? The viral DNA and the bacterial DNA. Suppose this is your virus particle and this is your viral DNA. Clear? And after attachment, they penetrate. Okay, and the viral genome insert within the host cell. Okay, and this is suppose bacterial DNA. Clear? And in case of lysogenic cycle, upon insertion, the viral DNA get integrated. Okay, integrated rather the disruption. That means instead of disrupting or degrading the bacterial DNA immediately, they form one association where the viral DNA gets integrated with the bacterial DNA. Clear? And this integrated condition is actually the prophase. Okay, and this prophase only is found in the lysogenic cycle. Okay, and in the latter steps, the prophase, after the prophase formation, induction process happen, And after in the induction process, these two get separated, and again they insert with the lytic cycle. Okay, and this is a brief intro regarding the lytic cycle of fire. Thank you all.